Analytical Chemistry 1, Lesson 35. Consider a solution with a weak acid in it at a very low pH, that is, a very acidic solution. If you begin to raise the pH of the solution, the fraction of that species that is present in its fully acidic form begins to decrease as the other, less acidic forms begin to appear in the solution. At any pH, it is sometimes very important to be able to identify the fraction of each species that can be present. A simpler but equally useful observation to make about the solution is answering the question, at a given pH, which of the possible species is the dominant one? Consider first a monoprotic acid, such as acetic acid. Its conjugate base is the acetate ion. At pH 4, what is the principal species present? Well, the answer is very straightforward. Take a look at the expression for Ka, or better yet, look at the Henderson-Hasselbalch equation. In the base form, the acetate ion, in this case, is equal in activity to that of the acid form, the protonated form. What does that tell us about the pH? Well, since they are equal, the ratio is 1 and the log of 1 is 0, and therefore the pH is equal to the pKa. The two forms of the molecule are equal in concentration when the pH is equal to the pKa. If the solution pH decreases, that is, the solution becomes more acidic, then the acid form will become the principal species. But if the pH rises, becomes more basic, it is the conjugate base form that becomes the principal species. The answer to the question of which is the principal species is easy. If the pH is below the pKa, it is the acid form that dominates. Above the pKa, and the base form dominates. Consider extending this to a diprotic system. There is a Henderson-Hasselbalch equation for each value of pKa. Both equations are simultaneously correct at all times. One is just more useful under certain conditions. If pH equals pKa1, then that means that the fully acid species and the intermediate species have equal concentration. If the pH is below pKa1, it is the fully acidic species that dominates. Above pKa1, and the intermediate species is dominant. With the other Henderson-Hasselbalch equation, we note that at pH equal to pKa2, the concentration of the intermediate form equals that of the fully basic form. Lower pH than pKa2 and the intermediate form dominates. Above pKa2 and the basic form dominates. In a multiprotic system, the values of pKa delineate the pH at which the principal chemical species switches from one form to another. Here is a graphical way to envision which form dominates for a tetraprotic system, such as nitrilo-triacetic acid. Each region is separated from its neighbor by a certain pH equal to pKa value. The principal species found in each region is labeled in its center. The concentration of the principal species in two neighboring regions is equal to each other at this pH equal to pKa value. In the center of each region, where the pH is the average of the two neighboring pKa values, the dominant species is at its largest value. Interestingly, while the two neighboring species are present at very low concentration, they are nevertheless equal to each other. There are different analytical experimental techniques in which knowing the dominant species is important. Being able to control that by changing the pH is equally important. Chromatography and electrophoresis are separation techniques that hinge on being able to control the speciation through the management of pH. Being able to identify the principal species is a useful, quick and dirty assessment of the molecular situation in a solution. Sometimes that is all that is needed. However, with a little mathematics, we can provide an accurate measure of the fraction of each species available at any pH value. First, look at a monoprotic acid. There are two forms of the molecule, conjugate acid and conjugate base, the protonated HA and the unprotonated A-, for example. And of course, this will work equally well if we discuss a base and its conjugate acid BH+. There is a formal concentration F, which is the sum of these two species. It is a constant and doesn't change with pH. And there is the equilibrium constant. We will cast it in terms of Ka, the acid dissociation. The fraction of the total amount of substance present in, for example, the HA form is just the ratio of HA concentration over the formal concentration. Similarly for the fraction of the conjugate base form. Well, let's first work towards the HA fraction. Rewrite the formal concentration and solve for A-. Substitute into the Ka expression. Expand and solve for HA. Substitute into the equation defining the fraction of HA. F cancels out, and the fraction in the form of HA depends only upon Ka and the hydronium ion concentration. It doesn't depend upon the formal concentration. We could get the expression for the conjugate base by following the same process of solving for A- instead. However, we can get right there by recognizing that in this instance, the fraction that is A- is the fraction that is not Ha. The fraction also only depends upon hydronium and Ka. These are the two key equations for a monoprotic species. Look carefully at the structure of these equations. Here's a graph that shows the fraction of the conjugate acid and base forms for two molecules, 2,4-dinitrophenol, which have a pKa of 4.114, and trimethylamine with a pKa of 9.799. First is the conjugate acid of the phenol compound. At low pH, it is the principal form, but it drops to 50% at the pH equal to pKa, and then continues down and is negligible at higher pH. The conjugate, the phenolate, starts off with no presence, but rises to 50% where the pH equals the pKa. It continues to become the dominant form at all higher pH values. In the case of trimethylamine, the ammonium form dominates at low pH and starts to drop as the pH approaches its pKa value, and the amine rises to dominate at higher pH values. Note how the curves for the two forms cross each other at 50%, and the crossing occurs at exactly when the pH equals pKa. So let's extend this to a diprotic molecule. There are three species. We label them H2A, HA-, and A2-. They can have different labeling, of course, if you've got an amine or something, but the pattern is the same. There are two acid dissociation equilibria with two pKa values. And there is a mass balance equation that equates the sum of the three species to the formal concentration F. As before, the fractional composition is the concentration of each species against this formal concentration. Let's first look at the fraction in the form H2A. Solve the two equilibrium equations for HA- and A2-. Substitute into the mass balance. Solve for H2A and put that into the fractional composition equation. The F's cancel out. Multiply numerator and denominator by H plus squared to clear out the denominators in the denominator, and you have the final expression. We can do something similar for the other two species. Without working through the math, here are the results. Take some time to look at the structure of these equations. Can you see the symmetry involved? Again, notice how the formal concentration has canceled out. The equations only depend upon the hydronium ion concentration and the acid dissociation constants. Compare them with the monoprotic equations we looked at before. And here is a graph of the three equations as a function of pH. The crossing points of the curves is at 50%, and these crossings also occur at the values of the two pKa's, 4.70, 9.97. In the pH range between 6.5 and 8, the intermediate form is pretty well alone in the solution. Now, here's an interesting graph. The molecule is now malic acid. It too is diprotic, but its two pKa values are much closer together, 3.459 and 5.097. You can see what happens to the intermediate. Its window of dominance is narrowed. The fully basic species starts to appear before the intermediate species gets a chance to fully establish itself. In the region around pH equals to 4, the intermediate species is still the principal species present, but there are significant amounts of the other two species present as well. This can be extended to systems with even more labile protons. Here are the equations for the four species involved in a triprotic acid-base system. Look carefully at the pattern. Hopefully, by comparing this and the diprotic case, you would be able to write down the equations for a molecule with any number of acid-base sites. Here's the fractional composition graph for phosphoric acid, H3PO4, a common inorganic acid. The fully acidic species dominates at low pH and drops quickly while the first intermediate species rises to dominance. It starts to fall as the second intermediate rises and then falls as the fully basic species rises to dominate at the high pH. Again, note how the curves cross at the 50% mark, located at the positions where the pH is equal to the various pKa values in sequence. And here are some additional examples of triprotic systems. First is glutamic acid. It is one of the triprotic amino acids. Note how its first intermediate doesn't get up to the top of the graph where it is essentially alone in solution. There's still about 8% of the fully acidic form and 8% of the next intermediate form in the solution when it peaks. Here's the graph for aspartic acid. It looks a lot like glutamic acid, just slightly different in some details. You might correctly conclude that the two amino acids are structurally very similar. Look up the two structures of these and see how they differ. That can explain why these curves are so similar. By contrast, here's another triprotic amino acid, cysteine. It is very different from the other two. Compare its structure and you can see why you would not expect them to be the same at all. Finally, here's the graph of citric acid. It is an organic acid, but it's not an amino acid. Both intermediate species are squeezed by the other forms, so that they never rise above about 75%. There is a small pH range where they are the principal species, but never a range where they are essentially alone. By comparison, the first intermediate for cysteine is the only significant species in the pH range of about 4 to 6. For aspartic acid and glutamic acid, their second intermediate species is the only significant species in the range of about 6.5 to 7.5.